The next hormone on our list is PTH, or parathyroid hormone. It is made by the chief cells in the parathyroid gland and is very important for regulating the balance between calcium and phosphate. Both of these play a significant role in bone homeostasis. The way I remember PTH is that it plays favorites. It likes to keep calcium around but doesn't want phosphate. So when you have high levels of PTH, you get hypercalcemia and hypophosphatemia. Some people like the mnemonic phosphate trashing hormone. So what's the mechanism of action? PTH will bind to the cells of the distal convoluted tubule and stimulate the resorption of calcium. Meanwhile, it binds to cells in the proximal convoluted tubule and it blocks the resorption of phosphate. That way you get more calcium retention and more phosphate excretion. Additionally, it acts on the kidney in another manner. Do you know what it does? It stimulates the activity of 1-alpha hydroxylase. And why is that important? Well, 1-alpha hydroxylase plays an important role in converting 25-hydroxyvitamin D into the active form 1,25-hydroxyvitamin D, or calcitriol. And vitamin D is really important for stimulating the absorption of calcium from the intestinal tract. That way, the calcium you get from your diet actually makes it into your bloodstream. We'll talk a little bit more about this later. Now, there's another way that PTH can raise serum calcium levels, and it is based on its activity on bone. PTH directly stimulates osteoblasts by binding to their PTH receptors. Do you know what happens as a result? It increases expression of rank L, or rank ligand. And rank L binds to rank receptor, which are found on osteoclast precursor cells, which stimulate the production of osteoclasts. And what do osteoclasts do? Right, they break down bone, which leads to the release of calcium into the serum. This process is known as bone resorption. Here's a diagram that illustrates all the effects that PTH have on the body. As you will see here, the regulation of PTH secretion is based on serum calcium levels. As you would expect, low serum calcium leads to increased PTH. Magnesium acts in the opposite manner of calcium, and if we decrease magnesium, we see a decrease in PTH. This is because exocytosis of vessels containing PTH require magnesium. This is unique because all other cells rely on calcium for the release of contents of their vesicles. Common causes of low magnesium include diarrhea or alcohol abuse and medications such as aminoglycosides and the loop and thiazide diuretics. Next, we'll briefly discuss calcium homeostasis. It is important to note that calcium exists in three forms, and only 45% of circulation is charged or ionized. The majority remains bound to either albumin or to other anions. This has important clinical consequences. For example, take this molecule of albumin. As you know, like most proteins, it carries significant negative charge, and hence it will be bound to cations such as calcium and hydrogen. Now, if a person were to have alkalosis for any reason, then the calcium would outcompete the hydrogen for binding spots in the albumin molecule. What is the consequence of this? Hypocalcemia. The patient may have cramps, paresthesias, and carpopedal spasm. So let's talk a little bit about vitamin D. Vitamin D is unique in that it is not only a vitamin, but it is also a hormone. There are two forms of vitamin D. Do you remember what they are? There is vitamin D3, also known as cholecalciferol, which is formed when provitamin D in the skin is exposed to sunlight. There's also a dietary form that comes from plants. This is known as vitamin D2, also known as ergocalciferol. Dietary vitamin D can also be found in the form of D3 when it is supplemented in foods like cereals, milk, orange juice, and both will be reabsorbed in the intestine and enter the bloodstream. Keep in mind that vitamin D is one of our fat-soluble vitamins. Remember ADEK? So patients with malabsorption, such as those with cystic fibrosis, will not secrete sufficient pancreatic enzymes and are at risk for vitamin D deficiency. So what happens to these forms of vitamin D? Well, they are taken to the liver where they become hydroxylated at the 25th carbon position. That's why we refer to these molecules as 25-OH vitamin D. Then what happens? Well, the 25-OH vitamin D becomes hydroxylated again. And do you remember where this takes place? Right, the kidney. And this time it's at the first carbon position, which is why we now call the molecule 1,25-dihydroxy vitamin D. And do you remember what enzyme is responsible for this? 1-alpha hydroxylase. And what hormone will upregulate the levels of this enzyme? Our friend PTH. Overall, both a functioning liver and kidney are necessary for the final production of active vitamin D. And you'll see many patients who have either liver or kidney disease, and these patients are at increased risk for vitamin D deficiencies. So what exactly does vitamin D do? Its main function is to increase the absorption of calcium and phosphate from the GI tract. It also helps to maintain calcium and phosphate levels in the blood. And overall, it is really important because both calcium and phosphate are required for bone mineralization. Do you know what is a consequence of vitamin D deficiency in children? Right, rickets. And what about in adults? We call it osteomalacia. We'll talk more about these conditions in the musculoskeletal section. Okay, time for a flash quiz. What form of vitamin D would you have to supplement in a patient with liver disease? If you're thinking 25-OH vitamin D, you'd be correct. Because you have a functioning kidney, shown here, you can still add the hydroxyl group to the first carbon. We've already talked about two hormones that increase serum calcium. But remember, our body likes to maintain homeostasis. So now we'll talk about calcitonin, which helps decrease serum calcium levels. Do you know where this hormone is made? In the parafollicular C cells of the thyroid. This is an important thing to remember because there is a subtype of thyroid cancer in which calcitonin is produced. Do you remember which one that is? Medullary carcinoma of the thyroid. It produces calcitonin, which is a great marker for this tumor. The function of calcitonin is to decrease bone resorption of calcium. We like to say that calcitonin tones down the serum calcium levels. But in actuality, this is not a very important role in calcium homeostasis, because patients who have their thyroid glands removed do not develop hypercalcemia. Throughout this lecture, we've talked about hormones and hormone receptors and the effects that these hormones have on the body as a whole. However, it's also important to understand the process by which hormones bind to their receptors and what second messenger systems are involved. In general, if you have a hormone that binds to a receptor, it may activate a second messenger molecule that can function to add phosphate groups to enzymes, which can either stimulate or inhibit their function and exert a given effect on a cell. But keep in mind, not all hormones use second messenger systems, and not all hormones bind to receptors located on the cell surface. Let's first start with the receptors. There are three major types that you need to know for this section. The first is the steroid receptor. This is a unique type of receptor because it is found inside the cell. What that means is that the hormone will have to cross the plasma membrane to reach its receptor. If that's the case, I'm sure you're thinking that these hormones are lipophilic. And you are absolutely correct. Examples of hormones that use intracellular steroid receptors include vitamin D, T3, T4, as well as adrenal steroid hormones including cortisol, aldosterone, and sex hormones such as estrogen, testosterone, and progesterone. The next receptor subtype is the intrinsic tyrosine kinase receptor. Let's break down the name. A tyrosine kinase is an enzyme that adds phosphate groups to tyrosine amino acids, and intrinsic implies that it does this without relying on any other molecules to mediate this action. This is known as autophosphorylation. Unlike steroid hormones, this receptor is located on the cell membrane. Hormones that bind to this type of receptor include insulin, IGF-1, and your other growth factors such as fibroblast growth factor, platelet-derived growth factor, and epidermal growth factor. The second messenger that is involved in the downstream signaling effects after this hormone binds to this type of receptor is the MAP kinase. The other receptor type is a receptor-associated tyrosine kinase. This is a type of receptor which cannot phosphorylate itself, but relies on a separate kinase to do so. In this case, it relies on the JAK-STAT signaling pathway. Here's an image depicting the JAK-STAT pathway. When the circulating hormone, shown here, binds to its receptor, it stimulates JAK kinase to phosphorylate the receptor as well as itself. Here you see the phosphate groups. This then allows for the STAT protein to bind to and become phosphorylated by JAK, as shown here. When there are two phosphorylated STAT proteins, they will bind to each other and go to the nucleus, where they bind to DNA and promote gene transcription. Hormones that use the receptor-associated tyrosine kinase include prolactin, growth hormone, and cytokines. There are also some other second messengers and their associated hormones that are important to know for boards. The way you might be asked about these second messengers is that they could describe a particular hormone and ask you to choose among the various second messengers which one would be associated with it. In this case, you may want to try using one of these helpful mnemonics to keep them straight. For cyclic AMP, you can use the mnemonic FLAT CHAMP to help you remember that FSH, LH, ACTH, TSH, calcitonin/CRH, HCG, ADH, MSH, and PTH all use this second messenger. You should also remember from your biochemistry lectures that glucagon uses cyclic AMP to activate pKa, which then phosphorylates hormone-sensitive lipase, leading to the mobilization of fat stores. The G-GOAT mnemonic will help you remember the molecules that use IP3 as a second messenger. G-GOAT stands for GnRH, GHRH, oxytocin, ADH, and TRH. Now, you may be thinking, wait a second. You already said ADH uses cyclic AMP signaling pathways. Now, what is it doing here in the IP3 signaling pathway? Well, it turns out that depending on what type of ADH receptor the hormone binds to, either V1 or V2, it will signal through either IP3 or cyclic AMP, respectively. Lastly, we have our cyclic GMP second messenger. This one is best to remember based on its function in vasodilation. The two major molecules that are associated with this are nitrous oxide, also known as endothelium-derived relaxing factor, or EDF, and ANP. And if you remember, ANP is released when your blood pressure is too high and is a potent vasodilator.